Amino acid and protein structure. We know that genes are lengths of DNA which code for protein, but we can break this down further. The DNA codes for the messenger RNA, which is made of triplet codons, each of which code for an amino acid, which in turn join together to make the protein. The amino acid is therefore the building block of proteins. There are 20 different amino acids, but they all share the same basic structure. The carboxylic acid group here confers the acidic properties, whilst the amine group here confers the basic ones. These are the same in all amino acids. This group here, the R group, is the variable region. There are 20 different amino acids, and that means that there are 20 different R groups. The most basic is a simple H, glycine, but they become increasingly more structurally complex. To form proteins, the amino acids must join together. They do this through a condensation to form a peptide bond. The OH group from the carboxylic acid end and the H from the amine group join together with and water is eliminated. So you have the OH group and the H group joining together. So water is eliminated in a classic condensation reaction. A dipeptide is formed with the strong covalent peptide bond in between the two amino acids. As many amino acids join together, the protein will start to fold and form a 3D structure. There are 4 levels of structure. The primary structure is the first level of folding or structure, and it is basically just the order of the amino acids, which really means the order of the R groups. This is the interaction between the R groups that will determine the next levels of folding. The primary structure is in itself determined by the DNA and by the genetic code, and any change in this can lead to a change in the order of the amino acids, which can lead to a change in the final 3D shape of the protein. The next level of structure is the secondary structure, which is the first level of folding. It's stabilised by hydrogen bonds between the R groups and also between the backbone. There are 2 types of secondary structure. The first is the alpha helix, where the amino acid chain literally forms a helix like this, which is stabilised by hydrogen bonds between the layers of the helix. The different proportions of alpha helix and the next type, which is the beta-pleated sheets, will determine whether the protein is a fibrous protein or a globular protein. The beta-pleated sheet is a much more structural form. You can see that you have more straight chains of amino acids or polypeptides, which are then stabilised by high levels of hydrogen bonding between them. If you have a larger proportion of beta-pleated sheet to alpha helix, then it would be a fibrous protein. If you have a larger proportion of alpha helix to beta-pleated sheets, it would be a globular protein. The tertiary structure is in many ways the most important. It is the final level of folding, and it gives the specific 3D shape. It is stabilised by 4 types of bonds: the hydrophobic hydrophilic interactions, the hydrogen bonds, ionic bonds, and covalent bonds or disulfide bridges. The strength of these bonds increases as you move down here, so the hydrophobic hydrophilic interactions are the weakest, and the covalent bonds, the disulfide bridges, are the strongest. Now, because this is a disulfide bridge, it can only occur between sulfur atoms, and so it can only occur where you have an amino acid that contains a sulfur in its R group. These are very, very strong bonds. The final level of structure is the quaternary structure, which only occurs when there are more than 1 polypeptide chain in the protein, and this will join the 2 polypeptide or 3 polypeptide chains together, such as in hemoglobin or collagen. The quaternary structure is stabilised by these same 4 types of bond.